Welcome to this web lecture on muscle function. By this time, you should have brought yourself up to speed or refreshed your memory on the basics of the sliding filament model of muscle contraction. You should know what I'm talking about when I mention a sarcomere or a cross bridge, what actin and myosin are, the role of calcium in muscle contraction. If you're feeling shaky on any of those concepts, go ahead and stop this web lecture and go back and review all of those things. You'll need them to understand what we're doing here. In the course of that preparation, you learned that muscles contract in response to usually stimulation by a motor neuron, that firing of the motor neuron is all or nothing. The motor neuron either fires maximally or not at all, so there's no variation in the strength of the signals that the muscles are getting. You also would have learned that the muscle contraction of the individual muscle fiber is also all or nothing. It's going to either contract maximally in response to that signal from the motor neuron or not at all. So given all of this all or nothingness, how do we end up with the ability to control the amount of force that we're using with our muscles? How do we have these fine gradations of force? And that's going to be the major topic of this web lecture, this idea of graded force, the idea that you can either lay your hand down gently on your desk or you can slam it down with a lot of force to get somebody's attention using the exact same muscles. How do we do this? And this is going to be very much related to this idea of force-velocity trade-off that we've already started to talk about when it comes to lever systems. So we're going to build this chart as we go with force-velocity trade-offs in one column, and then this idea of graded force production. How do we modulate the amount of force that we're producing with our muscles in the other? And so the first part of this I want to talk to you about is the force-length relationship. So before we go into the force-length relationship and the kind of experiments that were done to elucidate this relationship, it's important to know the overall architecture, the basic construction of this organ called a muscle. So we're going to start way down at the very smallest scale with these myofilaments. So the myofilaments are those actin and myosin protein molecules that you learned about in the sliding filament model. These are the actual motor that's going to generate the force, also called tension when applied to muscles. We refer to muscle tension. That's the same as the amount of force the muscle is generating. And that's basically going to come about because of the interaction of these actin and myosin myofilaments. These myofilaments are bundled together in these structures called myofibrils, and a whole bunch of these myofibrils are going to be grouped together in basically what is the muscle cell, also known as a muscle fiber. So an individual muscle fiber is going to run usually the entire length of the muscle organ, and it's surrounded by the cell membrane, in this case known as the sarcolemma, if it's a muscle cell. One interesting thing to note about these muscle fibers, these muscle cells, is that they're multinucleate. So you see two nuclei here, but there are actually several nuclei just on the inside of the sarcolemma, outside of these myofilaments. So these muscle fibers, in addition to having the cell membrane, the sarcolemma, they're also surrounded by this layer of connective tissue called the endomysium. The endomysium. So it's these layers of connective tissue that you see as this white surrounding each of these muscle fibers. These muscle fibers are in turn grouped together to form a structure called a fasciculus. So a group of muscle fibers is known as a fasciculus, and each fasciculus is surrounded by its own layer of connective tissue called the perimysium. So the perimysium is connective tissue that surrounds each fasciculus. A fasciculus is a bundle of muscle fibers, each of which is surrounded by a connective tissue called the endomysium. So a whole bundle of these fasciculi is surrounded by another layer of connective tissue called the epimysium, which surrounds this entire organ called the muscle. So a whole bundle of fasciculi forms a muscle, and they're surrounded by epimysium. In addition to that, they're going to be surrounded by this layer of loose connective tissue called a fascia. So all of these connective tissues are going to come together and fuse at this end where they connect to the bone in the structure called a tendon. So a tendon is basically all of these connective tissues running all the way through all of these different levels of the muscle and fusing together. This is why you can't just sort of take apart a tendon and separate a muscle from the bone. You have to actually cut it because it's completely integrated within all of the layers of that muscle. So that tendon in turn fuses, it's continuous with this layer of connective tissue that surrounds the bone called the periosteum. All of these layers of connective tissue going all the way through from this endomysium surrounding individual muscle cells all the way through the periosteum is one continuous sheet of connective tissue. So now let's think about the force-length relationship and these experiments that were done to determine this. So the first experiment I want to talk to you about, and this is one that you read about in your book, these are experiments done with individual muscle fibers with all of those connective tissue layers removed. So just the motor part of that, just that active part with the actin and myosin myofilaments that are going to be forming these cross bridges and generating active force. So if you take all those connective tissues away, the experimenters took the single muscle fiber and they have an apparatus that holds it at a particular length and then stimulates it to contract. When it does that, this apparatus measures the force that's generated at each particular length. So what are we going to do? We're going to have our apparatus hold it at whatever this minimum length is, stimulate it, measure the force, have it hold it at a little bit longer length, stimulate it, measure the force, longer length, stimulate it, measure the force, until we get this nice force length curve for a single isolated muscle fiber. And this is what we find. We get a maximum amount of force. So remember, tension, when we're talking about muscles, is the equivalent of the amount of force that it's generating. At an intermediate length, it builds up from 0 at this minimal length up to a maximum and then tapers off again. Let's take a look at what's happening at the level of the sarcomere to explain this pattern. So at the shortened length, the sarcomere is fully contracted. There's full overlap between these actin strands, and they can't go any further. They can't slide any further past the myosin, so they can't generate any force at this length. There's just nowhere for them to go. If we look at this point of maximum force, what we see here is maximal overlap between these actin and myosin myofilaments. This means that the maximum number of cross bridges can be formed. The amount of force is going to be determined by the number of those myosin heads that are pulling at the same time on the actin. All of those little individual forces will sum to create the maximal total amount of force when you've got this maximum level of overlap between the actin and myosin myofilaments. If we look at the very end of this, at the longest fiber lengths, now we've pulled the actin completely out of this myosin array. Again, there's no overlap between actin and myosin. There's nothing for those myosin heads to pull on, and again, they can't develop any force. When we're looking at just the active component, just the result, the force generated by these actin and myosin interactions, we get maximum force at this intermediate length. Let's take a look at what happens if we do the same thing with a whole muscle, so with all of those connective tissues in place. So we're repeating the same experiment. We're going to hold it at a particular length, stimulate it, measure the force, hold it, stimulate it. And this solid line that you see running through here is what we measure for a whole muscle. So let's break this down and see what's happening here. So we see that the first part of this curve looks almost exactly like the curve that we saw for an isolated muscle fiber. So that's this dashed line here. And we call this developed tension or active tension. This is the force that is the result of the interaction, those cross bridges being formed between actin and myosin. Then at some point we start to deviate from that curve. And this is going to be the result of those connective tissues. Remember, that's the only thing that's different about this experiment. So we can actually do this experiment without actually stimulating it and find out if we just put it at different lengths, how much force is it going to generate. And this is what this line is here. This is measuring the force at different lengths without stimulating the muscle to contract. So this is completely without any muscle contraction, no generation of cross bridges. We start at this length, we start to see more and more and more force being generated just as a result of the elasticity in those connective tissues. This is not the muscle doing anything to create force. This is responding to basically the machine pulling on those elastic connective tissues. So we call this the passive tension. So this is just responding to the pull of the machine. When we add what the muscles are doing with their actin and myosin to what the connective tissues are doing responding to the pull, at every point along here, if we add those two curves together, we get this total tension, the combined effect of the active tension and the passive tension to get the total tension. So what we see is right here where the muscle starts developing passive tension, that's its resting length. That's the length that the muscle would be, just without anything pulling on it, without the muscles contracting. This is where those connective tissues are at their preferred length. Anything greater than that, they're going to start resisting and generating that passive tension. So with this total tension, what we see is that we can really maximize the force that this muscle can generate by tapping into that elastic property in the connective tissues and using this passive tension to generate more force in the muscle. So this is another way we can get additional force out of the muscle when we stretch it beyond its resting length. And so this phenomenon is also going to be related to what we call the force-velocity relationship within muscles. So we'll see that this is going to come into play both in this force-velocity trade-off and also in producing graded force production. So let's take a look at how these experiments are done. So I only want you to pay attention to this line here that's kind of arcing down from high to low. Ignore this hump line beyond the scope of what we're doing here. Don't worry about the mechanical power output. So this is a very similar experiment where the muscle is taken and the machine controls how fast that muscle can contract. The machine is going to determine how fast that muscle fiber is going to shorten, and then it's going to measure the force on the apparatus as the muscle is trying to shorten at its maximum shortening velocity. So we're controlling the speed and measuring the force. And so in this graph we have the shortening velocity as a ratio of its maximum shortening velocity, the force as a percentage of the maximum amount of force. And what we see is that at zero shortening velocity, what does that mean? That means basically that the muscle is not shortening. The muscle is not shortening. This is what we call an isometric contraction. So this is basically a situation where you are trying your hardest, say to lift a table that's actually too heavy for you to lift, but you're activating your muscles as hard as you can, trying as hard as you can to lift that table, but it's resisting too hard for your muscles to actually shorten. This is an isometric contraction. And so we get our maximal force production at the point where the muscle is not changing length. As we allow the muscle to move faster and faster and faster, less force is going to be generated until we get all the way down to this other end of the curve with the maximum shortening velocity, which is when the muscle is not loaded at all. It's free to just shorten completely unresisted, and we call this an isotonic contraction. So when it's at its maximum velocity. So remember that the machine is controlling the velocity, and this velocity is a vector quantity. It has a direction, and we're calling positive velocity the shortening direction. This machine can also lengthen this muscle. So if this is its resting length, we can pull it and have a negative velocity in this other direction, a lengthening velocity. And if we do that, what we find is this force continues to increase. So you can think of this as maybe you pick up a weight that's already elevated, that's just a bit too heavy for you, and you're trying really hard to hold it up, but it's weighing your arm down more and more. So the muscle is actually lengthening as it pulls your arm down because it's applying too much force. We can think of that as what's called a lengthening contraction. Your muscle is contracting, you're trying really hard to resist that external force, but your muscle can't generate enough force to resist it. And so it's going to lengthen at the same time as it's trying to contract. So the muscle is active. Those cross bridges are trying to form and shorten, but the external force is just too high. So we call this a lengthening contraction when the velocity, the shortening velocity, is less than zero. And so it's contracting while it's being lengthened by an external force. Remember, muscles can only shorten forcefully, but if they're acted on by an external force, they can lengthen. We can maximize the muscle force by pre-stretching with an opposing muscle. So we're going to get greater force if we can actually have a lengthening contraction by using some opposing muscle or some opposing force to get that muscle to stretch while it's contracting. And we can see an example of this if we look at the wingbeat cycle of a flying bird. So the illustrations show you the position of the wing at different points during the wingbeat cycle. And these bars inside the circle are electromyography recordings of the muscle activity. So basically, they've implanted recording electrodes into the muscle and measuring when that muscle is activated. The pectoralis is the main downstroke muscle. The supracoracoideus is the main upstroke muscle. So remember, we saw that the pectoralis is the adductor. In birds, the main abductor is a muscle called the supracoracoideus. We'll learn much more about that muscle as we go. And so this is the point at which that wing begins to adduct. So the muscle starts shortening at about this point. This is the top of the upstroke. But what we see is that the muscle starts to be activated well before that, about 2/3 of the way through the upstroke. At this point, the supracoracoideus isn't even activated. It only needs to get that upstroke started. The aerodynamic forces pushing the wing up take over from there, and the pectoralis starts to be activated to slow down that upstroke, to resist that aerodynamic force pushing the wing up. But this pectoralis muscle is actually being lengthened at this point when the wing is still going up. So it's activated and lengthening. We've got a lengthening contraction that's going to maximize the amount of force that that muscle can produce to generate a very powerful downstroke. We can also look at the way the muscle is put together, the muscle architecture, to tell us something about these force-velocity trade-offs and to optimize for either force production or velocity production. Let's take a look at the way this works. So the muscle architecture is the overall shape of the muscle. So we can have parallel fibered muscles in which the muscle fibers are parallel to their tendinous insertion. They go right into the tendon, and the tendon continues in a straight line attaching to the bone. In pinnate muscles, these are two alternate spellings of this word. Don't worry about that. Either is fine. In these pinnate muscles, the muscle fibers come in at an angle to the tendon that they attach to. They aren't pulling directly straight down on their muscle. They're pulling at an angle. And one of the defining features of these muscles are their physiological cross-sectional area. So what do we mean by physiological cross-sectional area? So for a muscle, what this means is that if you were to take a scalpel and make a nice clean cut perpendicular to the muscle fibers, in this case right across there, perpendicular to the muscle fibers in this pinnate muscle, then if you took that cut edge and, say, photographed it, measured the cross-sectional area using computer software, that would be the physiological cross-sectional area of the muscle. It's the surface area of the cut edge if you cut through these muscles perpendicular to the muscle fibers. And this physiological cross-sectional area determines the amount of force that a muscle can generate. The larger the physiological cross-sectional area, the more force. So if we think about these cartoons of differently shaped muscles, we can see that in terms of force production, all of these are going to be the same. Even though this one has a lot more muscle mass, this longer one, it's still going to generate the same amount of force. So force equals force equals force in all three of these because they all have the same cross-sectional area, the same area of this cut edge. This one is shorter than this longest one, but it's going to have a greater amount of force because of this larger cross-sectional area. If we think about the velocity, the shortening velocity is going to be determined by the total length of the muscle. So now if we compare these muscles of different lengths, let's assume that these have all been cut exactly in half, and the lengths you see are proportional to their total length. If we have sort of the font size here reflecting the shortening velocity, the shortest one is going to be the slowest. These two will be both medium, because they're both the same length. So this fat one and this one in the middle, and then this is going to have the greatest shortening velocity. Let's take a look at why that is. So if we think about these parallel fibered muscles and pennate muscles, we look at the fiber lengths. Generally in vertebrate muscle, all of the sarcomeres are going to contract simultaneously, and they're going to shorten about 30% of their length. So if each sarcomere shortens simultaneously to about 30% of its length, that means that the whole muscle is going to shorten about 30% in the same amount of time. It's all going to happen simultaneously. So this parallel fibered strap muscle is going to shorten a much greater distance, because 30% of this longer length is going to be a greater distance than 30% of the shorter length. It's going to shorten a greater distance in the same amount of time, so it's going to have a greater shortening velocity. So the shortening distance and the shortening velocity depend on the number of sarcomeres in series. Why? Because each of them are going to shorten 30%, and so the more of them you have, the greater the total shortening that you're going to have. In contrast, the force generation depends on the number of sarcomeres in parallel. How many sarcomeres have individual little connections to this tendon? Each one of those sarcomeres pulling on this tendon is going to add its own individual force together to determine the total amount of force pulling on this tendon. So the number of sarcomeres in parallel is going to determine the force. The number of sarcomeres in series, how many sarcomeres you have lined up one after another, is going to determine the shortening velocity. So an example of some of these parallel fibered muscles are the strap muscles that we see in the necks of mammals. So you can see there are a great number of them. So here we have many sarcomeres in series, which enhances the velocity and shortening distance of these muscles. We can have another kind of parallel fibered muscles that are sort of a hybrid design. You kind of get a nice compromise, best of both worlds solution. These can tend to be fairly long. So here we see the biceps muscle and the upper arm. You can see the two heads of the biceps here fusing together into a single muscle. And so what we have here is a pretty long muscle with a lot of sarcomeres in series. It's going to increase the shortening velocity and shortening distance, but you also have this belly of the muscle. And so this is the typical shape that we think of muscles having, with this fat belly, and that maximum cross-sectional area through the middle of the belly is going to give it a large amount of force generation. So we call these fusiform muscles. An example of a pennate muscle is the gastrocnemius and the calf of humans and other mammals. And so here we see many, many, many sarcomeres in parallel. If we're going to cut perpendicular through all these muscle fibers, we pretty much start up here and go all the way around. And if you add together again all of those sarcomeres having their individual pulls on the tendon, you get a huge amount of force out of these pennate muscles. Another way the muscles can be arranged to be able to moderate force is the use of antagonistic and synergistic muscles. So synergistic muscles are two or more muscles that have the same action at the same joint. So if we look at this example in the cat, if we look at the pecto antebrachialis, which goes from the sternum out to the forearm, and the pectoralis major, which goes from the sternum up to the head of the humerus. These are both going to be adductors of the shoulder joint, and they're going to work as synergists, producing the same action. So one cool thing that we can do with these synergistic muscle groups is that even though they have the same action, they can be optimized for either force production or velocity production using leverage. So here we see a pair of synergistic muscles. One has its insertion more proximally, one has its insertion more distally. This proximal insertion is going to be optimized for speed. This more distal insertion can be optimized for strength. So we can see the speed component of it right here. The same amount of muscle shortening is going to cause a very large excursion as a result of this muscle. The same amount of muscle shortening, the same percentage of shortening, is only going to raise the one with this distal insertion a small amount. So again, if that happens in the same amount of time, this is going to be a higher velocity movement. And if we just fill in our levers here, as we've been learning how to do, we put in the line of action of this first muscle with the proximal insertion. Imagine that we are lifting some sort of weight out at the, I suppose it would be hand, although it's kind of hard to tell with this illustration, but let's say that we're resisting gravity, and so it's going to be a vertical line for the line of action of the output force. The perpendicular line to the center of rotation of the joint is going to be very short in this case, and then that perpendicular line to the output force is going to be very long, so we can say that this is going to amplify velocity at the expense of force production. If we look at this other one in the same way, here's the line of action of the muscle force with this more distal attachment. Again, we're resisting something at the end of this limb against gravity, so vertical output force. We draw in those in levers. We've got a much, much longer input lever in this case, the same output lever, so we can see that this one is much more optimized for force production. Conversely, antagonists are muscles that have the opposite action at the same joint, such as flexors and extenders. So if we think about the biceps and the triceps, those are a classic example of antagonistic muscle groups. And so if you are flexing at the elbow and you're not lifting any kind of weight, your hand is empty and you're just going to flex at the elbow, but you want to do it slowly, basically that biceps, when you tell it to go, if nothing's opposing it, it's going to shorten at its maximal velocity, right? Remember, zero force gives you maximum velocity, we saw in that force-velocity relationship. So what do you do if you want to just flex your elbow very, very slowly? Well, basically what you do is you simultaneously activate the triceps muscle to oppose that action and slow it down. And you can regulate the speed by using these antagonistic muscle groups. Another way you can get greater or lesser force in movements is by recruiting a larger or smaller number of motor units. So what is a motor unit? A motor unit is defined as a single motor neuron and all of the muscle fibers it innervates. So remember, the single motor neuron is going to either send a full signal or no signal at all. All of these muscle fibers are going to, when they get the signal, contract maximally. You can have different sizes of motor units. You can have different numbers of these axon terminals coming from the same axon. So you can have a small number of motor units with a large number of muscle fibers in each. So this would be a bunch of these little axon terminals coming from the same axon. And you would have the situation in muscles where powerful gross motor movements are needed. So you can think of this as being the large quadriceps muscles in your legs are going to tend to need to produce a lot of force for the kinds of movements that we see in walking and other gross motor tasks. Or you can have a large number of motor units, and each neuron is just going to stimulate a very small number of muscle fibers. And you would have this situation when very precise, fine motor control is needed, such as in the fingers, in the lips and tongue when you're talking. It gives you very fine control over movements. When more force is needed, more motor units can be recruited into that motion. When less force is needed, you can use a smaller proportion of that total muscle mass and recruit fewer of these motor units. The last mechanism for graded force production I want to talk to you about is something called multiple wave summation, or temporal summation of different nerve stimulations. So let's take a look at how that works. So to understand this, let's first think about the way a muscle fiber responds to a single neural stimulation. So most muscles are what we call twitch muscles, and in response to a single stimulation, they're going to produce a single twitch-like contraction that is going to look like this. So if this is time, the stimulation occurs at a particular point in time, and this is the amount of force that the muscle is generating. There's going to be a slight latent period, and then it's going to start to contract. It's going to reach a maximum tension and then go into a relaxation period where the muscle returns to its relaxed state. So you can think of this as there's going to be a burst of calcium deposited into the cell. It's going to get this actin and myosin interacting, forming cross bridges, but less and less calcium is going to be available when you get up to this point. It's going to be taken back up into that sarcoplasmic reticulum. As less and less calcium is available, those cross bridges are going to stop being formed and stop being reestablished, and then you'll go back into relaxation. But if you stimulate it more than once, so let's say you stimulate it a second time just as it's beginning to relax, you'll get something that looks like this. So basically you stimulate it, the calcium's released, those cross bridges start to form, you get more and more tension, the calcium starts to be taken up, but then you get another stimulation with another release of calcium. You can actually increase the amount of force that's generated because you keep that calcium in the cell environment for longer. You can make more and more cross bridges, and they can shorten to a greater extent and reach that higher level of force. So again, you got two stimulations here. The second one occurring before muscle fiber is fully relaxed. You can get an increasing amount of force. If you continue to stimulate the muscle fiber without allowing it to relax at all, this is where you get the maximum amount of force, and these little humps will fuse together if you do it fast enough, and we call this tetanic fusion of twitches, so you get a maximal force with continued stimulation of that muscle fiber. So this is the point at which there's plenty of calcium available. Cross bridges are going to be being made continuously as long as that calcium is available. A high frequency of stimulation is going to lead to this phenomenon that we call multiple wave summation. So we get multiple waves of stimulation. They're going to sum together to reach this maximal level of force at this point called tetanus. We've seen a number of different mechanisms that produce this force-velocity trade-off in muscles, some of them intrinsic properties of muscles, some of them come about in terms of the way the muscle is put together and attached to the skeleton. We've also seen many ways in which we can control the amount of force that is produced by our muscles. So let's just sum up. So although contractions of individual muscle fibers are all or nothing, the function of whole muscles can be modulated to produce graded force and optimize for either force or velocity, or sometimes a compromise between the two. So both of these can be done by exploiting the intrinsic properties of muscles, such as their length-force relationship, which remember is a consequence of the overlap between actin and myosin, myofibrils within the sarcomere, their force-velocity relationship, which has to do with opposing forces not allowing those cross bridges to generate their pull at quite the velocity that they would if they were being unopposed by a force, and their ability to sum individual twitches together to generate greater force in that multiple wave summation. They can also be done using the structural arrangement and architecture of the muscles by taking advantage of leverage, muscle shape, motor units, and these antagonistic or synergistic muscles to produce either more force or less force.